Welcome everyone. I am Mara Parvulescu. I will be your host today. I just want to welcome you to the webinar. I would like to say a few words first about MDPI. As you may know, MDPI is a pioneer scholarly open access publishing, and MDPI has supported academic communities since '96. We are based in Basel, Switzerland, and our mission is to foster open scientific exchange in all forms across all disciplines. We have more than 400 journals, at the moment 422. They are diverse and open access journals, also including 413 peer-reviewed journals. Also, we have 9 conference journals. We are supported by more than 1,015 academic experts who share our mission, values, and commitment, providing high-quality service for authors. About the journal LAND, because this webinar is made by the LAND Journal, we are international and cross-disciplinary peer-reviewed open access journal on land system science, landscape, soil, and water, also urban study, land climate interactions, water energy, land food, wolf nexus, biodiversity research, and health nexus, also land modeling and data processing ecosystem services, and multifunctionality and sustainability. We are published monthly online by MDPI. Our program for today will be, in the first part, we have a chair introduction, then Professor Nicholas Comerford, who will have his presentation. In the second speaker will be Dr. Zhao, followed up by Paola Green. At the end, we will have a Q&A, and we will conclude the webinar. What I would like to ask each participant is possible in the Q&A form when they will ask the questions to just let us whom they are and the question for who will be addressed. It's very important for us to know these details. Our webinar is based on the special issue challenges and solutions for sustainable land, soil quality, and contamination. The speakers for today are also the guest editors for this special issue, and the deadline for submissions it is next year in 30 June. Here you will see how the Q&A box is looking. And I would just like to tell you a few words about Professor Mink. He's emeritus professor and was affiliated to University of Florida, USA, and he's our webinar chair. Regarding Dr. Paola Greni, she's affiliated to Water Research Institute and National Research Council, and also she's one of the speakers. And Dr. Zhao, she's from Center of Crop Science in Queensland Alliance for Agriculture and Food Innovations, the University of Queensland. At the end of my presentation, I just want to present you a virtual event that the LAND will host. It is the second International Electronic Conference on Land, and it will be organized in 4 and 5 September next year in the Central European Summertime, and it will be online. We also left here a QR code that can be scanned by each participant. I will just give the word now to Professor Nicholas, and he will say a few words for the participants. Thank you very much.
Good morning. Let's see. I just need to share my talk. Okay, well, great. Then we're off and going. I'd like to say good morning. I guess I should say good morning, good afternoon, or good evening, depending on where you are, because we are going around the globe with this particular webinar. What I'd like to do is tell you what we're trying to do with this webinar. We have this special issue going called Challenges and Solutions for Sustainable Land, concentrating on soil quality and soil contamination. What we're trying to do with this webinar is we want to promote the special issue, and we want to solicit manuscripts. We want to solicit manuscripts that are going to address how soil contamination affects soil quality and sustainability. And we'll go a little bit through what we consider to be soil contamination, what would be like reasonable topics to deal with. That includes remediation strategies for contaminated soils. And we also want to provide with this webinar an insight into the range of topics that might be covered under this umbrella. So as we go through this webinar and all three programs, we're asking you to think about what kind of work you're engaged in and whether that work would be appropriate for submission to this special issue. Okay, let me see, if I can get this going. There we go. So what we have here is, excuse me while I try to get one thing here. Can I get this over here? There we go. This is going to be, okay. This is going to be 3 presentations as was suggested originally. The first one will be mine, and I'm going to talk about what the special issue it wants to cover and what might be appropriate for it, and looking at sustainability as defined by soil quality, which is based on soil ecosystem services. So we'll go a little bit of review what are soil ecosystem services, what that we mean by soil quality, and how those affect sustainability. The next talk is going to be by Dr. Chow on sensing technologies and characterizing and monitoring site quality and soil functions. And you'll see where the soil functions component comes into talking about sustainability. And then this will be brought up, well, batting cleanup, as we would say in the United States, is the importance of microbial communities in soil quality, ecosystem services, and the remediation of contamination. This is Dr. Grenny, and she is going to bring, I think, all of that together from a microbial perspective. So this is sort of the basis of how I look at this special issue and what we're trying to do. Soil contamination is going to affect soil ecosystem services. Soil ecosystem services help define what is soil quality. And soil quality is a major determinant in the sustainability of soils. So if you think about soil contamination, it's an addition to the system. The soil ecosystem services are going to describe the processes within the system that are going to determine what is soil quality. Soil quality is really a product from ecosystem services, and we usually think of it in terms of specific indicators to try to identify the quality. And that quality then is a determination of whether an area will be sustainable or not sustainable. So that's sort of the basis of where we're going over the next couple minutes. So soil ecosystem services, to get us all on the same page, reflects the benefits that soils provide to humans and the environment through their natural functions and processes. Interestingly, it was originally defined just as the benefits to humans, but I think over time it has developed also looking at the benefits to the environment, and this is an advance. This is a step up from what the original definition was. Soil ecosystem services then are divided into a variety of different categories of services. First one being provisioning services. These are the products that come from ecosystems. Food, fiber production would be one. Soils support the growth of crops. Soils support the growth of trees. And then with forages, the forages also support livestock as a product, as an ecosystem service. So as you're thinking about this and what your work is, these are the potential products or provisioning services that might be appropriate, looking at how contamination could affect them. Raw materials are another important component that product from soils. Clay, sand, and gravel used in construction. Another real important product that you get from soils are pharmaceuticals or medicines. The most obvious, or the most well-known one, would have been penicillin. Another one would be streptomycin. It's been estimated that there are tens of thousands of potential medicinal products that still could be developed from soils. And that's because there are an enormous number of microbial species that have yet to be identified in soils. and the advantages that soils have given in terms of biodiversity are huge. We often hear the phrase that above ground biodiversity is just a footnote to the kind of biodiversity that you find underground. Another ecosystem service on the provisioning end is water. Soils supply water. They allow water to infiltrate. They're stored as groundwater. And then these are crucial products for both drinking water and irrigation. In fact, here in the United States, if you think about the Rocky Mountains, there's at least 30,000 cities in our mountains that get their water solely from water that passes through the soil in the mountains and comes down. Another group of ecosystem services are the regulating or supporting services. When we talk about contamination of soils, this is really, I think, where the focus is, because we're thinking about contamination affecting processes, and the regulating services are just a set of processes that are defining how a soil works. Examples, and not a complete set of examples, but examples would be nutrient cycling. Soils facilitate, we know, the decomposition of organic matter and the cycling of nutrients. If you're dealing with soil nutrient bioavailability and the effect of contaminants on that, that might be an appropriate kind of a topic. Another regulating service, we talked about the importance of water. Water regulation is an incredibly important component because soils regulate the water flow and help in controlling flooding. Why is that? Because soils are porous. Soils have the ability to hold water once it gets in there, and it has the ability to release water. And how soils release water to, say, rivers determines the water flow in many ways. So regulating services are important. Another regulating service is climate regulation. The concerns on climate change are all about sequestering carbon dioxide, sequestering carbon. Soils are thought to be one of the largest potential solutions to how to sequester carbon and lower carbon in the atmosphere because of the amount of land service with the proper kinds of management systems. The potential is there. Whether it's there for the long term or not is another question. Whether it's truly sequestered or not, another question. But how contamination affects soil sequestration of carbon is an important issue. Pollution filtration. Well, obviously, this is directly related to this special issue. How soils filter pollution, how they break down pollutants when they get there. And I think that's going to be an important consideration when you get to Dr. Grenny's presentation about how microbial communities can break down different pollutants. These include things like organic pollutants, pesticides, heavy metals, a lot of work done on heavy metals in the past, and all kinds of waste products. Again, habitat provision. Soils offer habitat for a wide range of organisms, and it's this living community in the soil that truly defines what a soil is. And again, we'll get to Dr. Grenny's talk at the end where she's dealing with microbial communities and what they can do. But these affect what the soil food web is, and how contaminants can affect this. We'll give some examples a little bit later. But habitat is probably one of those key ecosystem services that are related to this special issue. Cultural services. This is something we don't think about very often, but it's one of the things that we see every single day as we go about our daily lives. Recreation and particularly aesthetics. I don't know what it's like in all your countries at this point in time, but we're in fall here in Eastern United States. It's the time when leaves start changing color in the hardwoods. As a forester, my background is in forestry. That to me is the perfect time of year, where the landscape is just robust with colors as leaves change and as the seasons change. Contaminated soils can very much affect that. So aesthetics is an important cultural service. If you're looking at those kind of possibilities, maybe this helps. Recreation. Everything we recreate tends to be soils, right? Because of contamination, can we continue to use them for recreation or not? There's many examples where that's not the case. Then we also talk about historical battlefields, and this is Arlington Cemetery here in the United States, where it's a culturally important area for us because it represents the soldiers that have given their lives for our country. Religious areas, particularly among indigenous communities, they have areas that are specific and highly sacred to their own communities, and we don't want to be able to affect those. How does soil contamination affect that? And then what we all know about is residential use. We live on top of soils, and our ability to live on those soils depends on the quality of the soil in terms of the health of the folks living on top of it. So that's just a brief smattering of what ecosystem services are. These are the processes that are going to affect soil quality. So let's define soil quality for our purposes. Soil quality is the ability of soil to function effectively within its ecosystem, supporting plant growth, maintaining environmental health, and sustainability. So there's a connection between soil quality and sustainability. It encompasses physical, chemical, and biological properties of the soil that influence its overall health and functionality, which is sustainability in many cases. We usually talk about soil quality more as a product than as a process because we use indicators to determine whether soil quality is high or low or compromised. Things like soil structure, compaction might destroy soil structure. Nutrient content, erosion might remove nutrients from the system. pH level, organic matter, huge. Organic matter is a huge one in terms of whether a soil has sufficient quality or not. And then we always come back to water one way or the other because water is an incredibly important capacity that the soil provides for ecosystems and for human health. And then we get back to Dr. Grenny again on biological activity. So now that we've got a little bit of ecosystem services and its effect on soil quality, what are the kinds of soil contamination that might be appropriate in your work that would lead to a submission to this particular special issue? I've highlighted 4 up front just because this is my own personal view. These are areas that I think need to be looked at in more detail. Radioactive contamination. Well, in Japan, there was a tsunami some years back that compromised a nuclear facility. In Ukraine, you've got Chernobyl. And then with all the wars going around the world, there are some possibilities of compromising nuclear power plants. But that's not just only one of the possibilities of radioactive contamination. I think there needs to be much more done in this area. Plastic and microplastics. This is something that has come along with our development as a civilization over time, how this is affecting soils. Pharmaceuticals. Big area now in terms of particularly using wetlands, soil wetlands to detoxify pharmaceuticals. Salinization. As we use more and more water, we have the possibility for saltwater intrusion and salination of our soils. How do we overcome that? And there's been some interesting work done recently. And then there's the normal things that we've seen done for decades, heavy metal contamination by applying waste to soils, pesticide contamination using pesticides in agriculture. Nutrient overload and eutrophication has been around with us for literally decades as agriculture chemicals have leached into the water. Acidification, you remember during the 80s, acid rain and the effect that acid rain had on soils, and then just all kinds of industrial and urban waste contamination, asbestos being an example. So what are the effects now of contamination on soil ecosystem services? This is really what we're trying to get at in our special issue. Just some quick examples. Reduced fertility and food production might be how contamination is affecting these ecosystem services. The impairment of biological processes in the soil that support plant growth are going to be compromised. Back to water regulation. You'll see that clogging of soil pores is a potential effect of adding contaminants to the soil. Carbon sequestration, as we mentioned before, really, are contaminants affecting our ability via the effect on microbial communities? And then I always get back to habitat for biodiversity because this is such an interesting area in soils. Reduction in biodiversity disrupts function and threatens entire food webs in the soil. So now what about the effects? So that's the effect on soil ecosystem services. What are potential effects on soil quality? Alteration of physical properties, we mentioned before, soil structure, soil porosity, and permeability, criteria used in soil quality, making soil more prone to erosion and reducing its ability to support plant growth. Altering physical properties is something that I think hasn't been looked at in more detail than it should be relative to contamination of soils. Chemical imbalances, if you alter the soil pH, you alter bionutrient availability. You disrupt the balance of essential nutrients, you can affect micronutrient deficiencies, as well as, I'll show you here in a second, toxicities. Reduce microbial activity. So microorganisms, as we've said, are essential for decomposing organic matter and cycling nutrients. So soil quality is affected by again, changing soil quality, changing the soil microbial communities and how that might be detrimental to the overall functioning of the soil. Toxicity to plants and animals. Number of these contaminants can be highly toxic. Arsenic is a good example we've had in the state of Florida, where we've had large amounts of arsenate applied to the soil because we used to use big pools of arsenate in the soil, and we'd walk our cattle through that pool of arsenate to kill ticks on the animal. And now we have arsenate, which mimics phosphate very nicely in soil chemistry that can be polluting the soil and polluting our water. Toxicity of plants give you poor growth, and animals rely on plants or to live within the ecosystem. They are going to face health risks. So the effect now of contamination on soil sustainability. We've gone contamination to soil ecosystem services, to soil quality, and now sustainability. And this is an example here, just as the effect on resilience. Resilience is the ability of a soil to recover from disturbance, right? A healthy soil can recover. And if you look at this graph here, what you're going to see is that soil resistance depends on the processes that go on in the soil. And anytime you compromise those processes, you are compromising the potential resilience of the soil. Loss of functionality, which is basically soil quality. Heavily polluted soils are no longer able to fulfill critical ecological roles. And we've talked about nutrient cycling, supporting vegetation, again, water flow. All of this leads to reduced sustainability in ecosystems. So we're trying to just push this whole idea of ecosystem services affect soil quality, affect sustainability. Degradation and desertification. This seems to be an increasing problem around the world, particularly desertification. Soil quality can decline due to contamination. In that case, they become more prone to degradation. Processes like erosion, compaction, desertification. These lead to the loss of arable land. These lead to long-term damage to ecosystems. And that's obviously going to reduce the long-term sustainability of any kind of a system. So that's basically a short review of what we're trying to do with this special issue. Challenges and solutions for sustainable land. I've talked mainly about the challenges that we're facing relative to soil contamination, and have not really talked much about the solutions. We're really looking for papers for your work, for you to submit to here, that deals with soil quality and contamination. In review, I think contamination is an input. It controls ecosystem services. Those ecosystem services that you really want to concentrate on, that we're looking to concentrate on, are the ones that are process driven, functionality driven, because those functions control the indicators that we use to determine soil quality. And that soil quality, as you've seen, if it's reduces the sustainability of a system, particularly if it reduces the resilience of a particular system. So with that in mind, what I'd like to do is introduce our next speaker. And that would be Dr. Chow. Dr. Chow is a research fellow in the Queensland Alliance for Agriculture and Food Innovation at the University of Queensland, Australia. Her research goal is to increase crop productivity through understanding how soil-crop interactions determine productivity. Right now, again, you can see directly what her expertise is to this special issue. She combines remote sensing and modeling with artificial intelligence to work towards her goal. She adds unique understanding to the topic of this special issue and how to evaluate the program. If we can't monitor, if we can't measure, then we have no way of dealing with contamination effects on soils. So with that, let me give you Dr. Dongxi Chow.
Hello, everyone. Today I'm going to introduce my research on digital soil mapping using proximal soil sensors for soil characterization and precision soil management. If you can't measure it, you can't manage it. As the world famous management consultant Peter Drucker once said. And as a soil researcher, I could not agree more. If we can't measure the spatial and temporal variations of soil properties, we cannot reasonably assess the quality of soil or engage in sustainable soil management. Traditionally, soil maps are developed by soil surveyors. It relies really on the expertise of the surveyors. And if you have two experts, you might get totally different results. Alternatively, soil maps can be generated based on the laboratory soil analysis results, but the lab soil analysis is quite expensive and time-consuming and unrealistic to be conducted when the soil variation is large, and especially when a large number of soil samples need to be characterized. So to bridge this gap, and digital soil mapping concept was proposed at around 2 decades ago, and which involves the creation and population of spatial soil data by the use of field and laboratory observational methods coupled with numeric or mathematical or geophysical models. So under the digital soil mapping concept, my study has focused on connecting easy and cheap to acquire the digital data, which could reflect the soil forming processes or soil forming materials with modeling techniques to generate accurate soil maps for applications in precision agriculture or in soil management in environmental science. And the digital data can either be sensed proximally or remotely, such as the drone or satellite imagery or the proximal sensed electromagnetic induction or the visible near infrared spectroscopy. This figure highlights the useful parts of the electromagnetic spectrum from the radio wave to gamma ray wave for obtaining the information on soil through proximal or remote sensing. And in the radio wave, the EM induction, the electromagnetic induction, the EMI, is one of the commonly used soil sensor in digital soil mapping. And in the visible and near infrared wavelength range, the optical sensors, such as drone or satellite imagery or NIR, has been used. And the gamma ray spectrometry, which can measure the radiation naturally emitted from the Earth's surface, is another important source of data for digital soil mapping. And this technique has also been applied to estimate the variation in the soil texture or the in the surface soil moisture content. Now, let's dive deeper to the applications of some of these sensing techniques conducted in my research. The EMI is one of the commonly used proximal soil sensor. And the beauty of this sensor is that it is contactless and can be towed by vehicle to quickly generate spatial dense soil information across large spatial scales. And the EM sensor, the principle of how the EM sensor works, including it shoots an electromagnetic pulse into the soil and record the soil responses. And we call this soil responses as soil apparent electrical conductivity, ECA. The ECA is a function of the conductors in soil, including salts, clay minerals, clay content, and also soil moisture. So in this case, we can convert the ECA map to high spatial resolution maps of these affecting soil properties through developing predictive models. And in the following several slides, I will show the several examples of the EMI application for predicting these soil properties that can reflect the soil quality. So first example. In the first example, the EMI was used to characterize the distribution of soil salinity in an irrigated cotton farm in New South Wales, Australia. And in this study, the EMI measurements were conducted across the farmer's field, as indicated in the yellow dots. And 20 soil cores, as shown in the red dots, were also collected, bringing back to laboratory for soil ECE analysis and also for developing a linear regression model between the measured ECE of these soil samples and the EMI readings. And with these 20 soil sampling points and the linear regression, and the authors expanded the relationship to the whole field and developed the soil ECE maps at different soil depths. And the predicted salinity maps on the right-hand side show that the eastern 2/3 of the field was characterized as extremely saline. And this zone also is the location closest to the water storage reservoir. So which means that the salinity problem in this zone might be a result of water leaking from the water storage. And these maps can help us to take targeted actions like variable rate gypsum application to address the salinity in this specific zone, and rather than treating the entire field. And also it can help us to decide where to install drains in the field to lower the water table and to reduce the salinity hazard in this field. In the second example, the EMI was used to map soil clay content across the Ashley irrigation area in New South Wales, Australia. And in this study, the EMI measurement was conducted across the area as indicated in the green dots. And in total, 40 soil cores, as shown in the red dots, were collected, bring back to lab for particle size fraction analysis, and also for developing a predictive regression between the clay content of these samples and the EMI readings. And we expanded the predictive relationship to the whole area and generated this three-dimensional soil clay map across the region. So this map is 40 kilometers wide, 25 kilometers high, and 8 meters deep. And in this map, the green color indicates a clay content larger than 40%, and the red color indicating a clay content less than 40. And you can see that this map can clearly indicate the spatial distributions of the prior stream channels or the paleo channels in this region. And we noted that a water storage dam here, which was known to be problematic, was situated on top of the sediments associated with a prior stream channel, which caused the deep drainage problem here. So this map, the application of this predicted clay map can be used to get the allocation of water storage reservoirs in irrigated cropping regions, and also can be used to reduce the deep drainage risk and to improve the water use efficiency in the region. In the third example, the EMI was used to predict the soil cation exchange capacity in a cotton growing field in New South Wales, Australia. And in this study, continuous EMI measurements were conducted along 19 research transects across the field. And in total, 25 soil cores were collected for laboratory CEC analysis and developing a linear regression model between the measured CEC of these soil samples and the EMI readings. And the linear relationship was then further expanded to the whole field and to generate the spatial distributions of the soil CEC across the field at each of the soil depths. And these soil maps can help us understand the soil fertility levels and manage this field more efficiently. And for example, in the areas with low CEC content, as indicated as light blue, the nutrients may leach faster and requiring more frequent fertilization to maintain the nutrients levels. And also by knowing the different CEC levels, we can also decide on the correct types and amounts of the amendments like lime or gypsum to adjust the soil pH or to remediate the soil acidity in the field. In the fourth example, the time-lapse EM surveys were conducted to image the soil wetting and drying cycle in a flood irrigation cotton field. And the EMI surveys were conducted along a transect from high ditch to tail ditch. And the first EMI survey was conducted before irrigation and commenced indicated at hour 0. And later, 6 subsequent EMI surveys were made during irrigation. And the irrigation stopped after 9 hours. And from here, the EMI surveys were conducted every day after irrigation until 11 days later. And from here, the EMI surveys were, and 8 soil cores were collected across this transect to calibrate the EMI readings to soil moisture content and to develop this soil moisture map. And this soil moisture map can help us to monitor the irrigation efficiency in this field and get us to schedule the irrigation at the right time and at the right location. And also, it can help us to maximize the water use efficiency in this irrigated cotton field. And recently, the proximal EMI sensing has also gained attention from the plant scientists for studying the soil and crop root interactions. It is particularly valuable for monitoring the crop water use in field agronomic research and also for screening genetic differences in water use efficiency in crop breeding trials. Okay, another rapid and cost-effective technology for soil characterization is visible near-infrared spectroscopy with NIR. And you can see me in this figure. I'm collecting the signal of the soil near-infrared spectrum using an ASD field spec4 spectral probe. And 1 second, 1 click, and 1 soil NIR spectral recorded. And this sensor can also be used directly in the field on bare soil cores, which can avoid the preparation of soil samples, which makes it more time and cost efficient. And the NIR spectrometer measures the soil spectra between 400 to 2,500 nanometer wavelengths. And the soil spectral characteristics in the NIR region are decided by the electronic transitions of atoms and the vibrations of molecular bonds in the soil, which makes this soil spectra encoding extensive information on the soil compositions. And by applying the data science and modeling techniques, we can extract the information of soil properties. Actually, it's multiple soil properties simultaneously from one single soil spectra. So how do we transfer this soil spectrum into valuable information of soil properties, either on the content of soil carbon or soil clay content or other soil properties? This comes to the soil spectroscopy data analysis. So after getting the soil spectrum, we need to do some spectral pre-treatment before we do modeling. It includes converting the soil reflectance to absorbance, filtering, trimming, and standardizing the spectrum to remove the background effects, and then using models to extract the soil information from the pre-treated soil spectrum. And partial least square regression is the standard model for analyzing spectrum data. And recently, machine learning models such as the regression tree, the Q-based, or the random forest have been widely used in soil science to analyze the soil spectra and to extract the soil properties information from the soil spectrum. And a recent study from Sydney Uni identified that 2,300 to 2,340 nanometers in the NIR range is highly correlated with the hydrocarbon contaminations in the soil, and which means the soil spectrum can be used to assess the petroleum contaminated soil. And if you are interested, you can refer to this study. What is the future direction? So each soil sensor has its own pros and cons. We could consider to use data fusion of multiple sensors coupled with robust model averaging techniques or machine learning models to further improve the accuracy and the reliability of digital soil maps. And if you are working on using sensing technologies to assess the soil quality or the soil contaminations, you are more than welcome to submit your study to our special issue. Thank you very much.
Thank you, Dr. Chung. I'll have to admit, I sit here amazed at how far the remote sensing has come in the last, well, I've been around a while. The fact that you can get to such depth, I think, is absolutely amazing and adds potential to looking at contaminated soils and long-term effect. Thank you for that presentation. It was excellent. I'd like to introduce our next talk, and this individual has a unique talents relative to this special issue. Dr. Paola Greni. She's a senior researcher at the National Research Council of Italy, an expert in microbial ecology of contaminated environments, so obviously central to what we're talking about here. An expert in ecotoxicology and bio-assisted remediation processes. So here we've got remediation solutions that we can consider. She has investigated a wide range of contaminants, including pharmaceuticals, antibiotics, and fertilizers, just to name a few. She's a well-known authority on the topic, and she is crucial to expertise in this special issue. So I would like to give you Dr. Paola Greni.
Thank you so much. Thank you for the introduction. Can you see my presentation? Okay.
Yes, we can see.
So today I would like to talk about the importance of soil microbial communities in soil quality, ecosystem services, and remediation of contamination. As already talked by Professor Nick, microbes have several functions and have, despite their small size, the natural microbial communities have a huge impact on our environment, providing several benefits to humans, called ecosystem services. Ecosystem functions provided by microbes are mainly the transformation of inorganic carbon into biomass, the nutrient regeneration and cycling, the conversion of organic matter, and the regulation of biogeochemical cycles. But also they have a crucial role in removing xenobiotics. Several bacterial strains have been isolated in our institute for their capability to degrade, for example, herbicides, biocides, chlorinate solvents, etc. So microbial communities provide ecosystem regulation services. Functions such as the degradation of organic matter require complex metabolic pathways, including the degradation of xenobiotics. And the recovery from the contamination is only possible if the toxicity of the xenobiotics does not hamper the microbial activity. For example, if there is a huge amount of contaminants. And the knowledge of the natural remediation capacity of a microbial community makes it possible to assess how much contaminants can enter higher levels in the ecosystem food web. Here you can see, for example, that a chemical can be degraded by, for example, chemical degradation or photodegradation, but the microbial degradation has a crucial role in the degradation because it is the only mechanism for which the degradation of a chemical can be to CO2 and water. And this is because the microbial communities have a higher physiological versatility and catabolic potential. So the presence of an abundant and varied microbial communities is a necessary prerequisite for an immediate and effective response to the various chemicals that can contaminate an ecosystem. Water and soil quality is regulated by microbial communities, so the ability of soil and water to recover from chemical contamination is primarily dependent on the presence of microbial communities with the ability to degrade and consequently remove it. So the effects of a contaminant on natural microbial community can be evaluated in terms of change in community structure, so the dominance or disappearance of some bacterial groups that can have some important ecosystem functions, and the evaluation of the presence of specific bacterial population adapted to the contaminant and able to use it as a source of carbon or nitrogen, selecting specific bacterial population able to remove and using the chemical as a source of carbon nitrogen. So the knowledge of ecosystem services provided by microbial communities is a crucial point for managing the recovery of contaminated water or soil. And although the potential homeopathic ability of microbial communities to remove contaminants is enormous, but it is not fully exploited. So let's go to the nature-based solutions. The nature-based solutions are action in harmony with natural principles to achieve development goals and addressing challenges. And they have recently gained popularity as an integrated approach that can address climate change and biodiversity loss, supporting a sustainable development. Using nature-based solutions, we can protect, sustainable, manage, and restore natural and modified ecosystem with benefits for both people and biodiversity. Traditional solutions can be modified by replacing hard infrastructure with greener environmental and offer ecological and societal benefits and helps combat flooding, erosion, extreme temperature, and restore biodiversity. Here I will present some examples of the application of nature-based solutions that highlight the importance of microbial communities in degrading contaminants or in recovering degraded soil. The first example is regarding the excavated soil from mechanized tunneling that contain foaming agents. In this case, we found a microorganism that were able to enhance the degradation of this contaminant, sodium lauryl ethyl sulfate. The second example is the soil contaminated by PCBs, that it is a very persistent contaminants and heavy metals in an area in south of Italy. And we use plants and organic amendments for recovering contaminated soil. And the third example will be the soil or sediment contaminated with persistent organic pollutants. And we use the microbial field cells for enhancing pollutant degradation. Excavated soil are soil that come from tunneling projects, and because there are several projects planned in Europe and worldwide, several hundreds of millions of tons of excavated soil have been continuously produced. Spoil material consisting soil and rock treated during the excavation process with foaming agents and polymers that are commercial products that can contain chemicals that are necessary for the modern tunnel boring machine. Some directive in Europe requires spoil material reused or recycled in accordance also with green economy goals. And debris can be used for filling old quarries or other places for green areas, for example. In accordance also with the Italian legislation, the material from the tunnel excavation can be used as a byproduct. if the limit of some organic and inorganic contaminants are not exceeded. Our recent works showed that the foaming agent can be toxic at high amounts for aquatic organisms, depending on their composition and manufacturing processes. The anonic surfactant sodium lauryl ether sulfate is the main component of several foaming agents used for mechanized tunneling. It can derive from petroleum hydrocarbons or vegetable tissues, and a prompt sodium lauryl ether sulfate biodegradation is a key point for the reuse of the excavated soil. Ecotoxicological studies performed on the most common commercial products made it possible to highlight the most environmentally friendly ones in terms of biodegradability and ecotoxicity. And because some commercial products resulted more toxic than others, some works stimulated chemical producers to design new eco-safe products. In our institute, we selected a bacterial consortium able to grow on sodium lauryl ether sulfate and to degrade it. The bacterial consortium was characterized, and it was in the gamma protobacteria group, and was able to degrade this contaminant in few hours. In the presence of the bacterial consortium, the sodium lauryl ether sulfate was degraded with a GT50 of 1 day. At the same time, in absence of this bacterial consortium, only the half of the amount of slash was degraded in a batch experiment. Some general conclusion of the bioaugmentation that we did. Bioaugmentation is a promising low-cost and natural-based solutions for a prompt and safe reuse of the spoiled material. Avoiding its temporary storage and necessary waste production and transfer to landfills. The overall results show how a diverse approach involving chemical and microbiological assessments of polluted soil can improve our understanding on the biodegradability of pollutants in bioremediation strategies. The bacterial consortium can also be used for remediating surface water from possible sodium lauryl ether sulfate contamination, being this anonic surfactant a common component of personal care products. The second example regards the soil contaminated with the PCBs and heavy metals. In this case, we use plants and organic amendments for recovering contaminated soil. Phyto-assisted bioremediation exploit the synergy action that is established in the rhizosphere between the root system of plants and microorganisms to remove, transform, or contain toxic substances in soil, sediments, water, and even in the atmosphere. Plant species through the rhizosphere modify the chemical physiological properties of contaminated soil, realize radical exudates, which stimulate the proliferation of microorganisms and can directly promote the degradation of contaminants. And microorganisms are mainly responsible for the degradation of the contaminants, since they are only ones able to mineralize the contaminant molecules. We study an area in the south of Italy near Taranto city, in which a chemical contamination by PCBs and heavy metals was found. We performed a microcosm experiment for testing a popular clone if it was adapted to this contaminated soil, and if the addition of compost was useful for enhancing PCB degradation. And you can find in these two papers what we found that was very useful, this popular chlore. Then we performed an open-air experiment using this popular clone, and we found a significant increase in soil dehydrogenase activity, which reflects a general improvement in soil quality that was highlighted by the increase in organic carbon content development. Organic carbon values in the rhizosphere have tripled. The values of microbial abundance and survivability have increased significantly, and the results of the heavy metal analysis indicates a phytostabilization process in soil, absence of translocation of pollutants. That is very important because in this case you can use the biomass, the poplars, as for other purposes. And PCB's analysis confirmed that this contaminant was degraded by the rhizo degradation. The third example regards the use of soil or sediment contaminated with persistent organic pollutant. In this case, we use microbial fuel cells for enhancing pollutant degradation. Terrestrial microbial fuel cells are bioreactors that transform energy stored in chemical bonds of organic compound in soil into electrical energy. TMC consists of anode and cathode compartments divided by a layer of soil, and these bioreactors use bacterial capabilities to oxidize organic and inorganic matter for the generation of electrical energy. And recently, microbial fuel cells attracted much attention as an energy-saving bioremediation approach. The exoelectrogen bacteria are key players in terrestrial microbial fuel cells because they metabolize organic compounds and transfer electrons to anode, then to the cathode, where oxygen usually acts as an electron acceptor. A microorganism associated to anode compartment from complex biofilms, able to transform a wide variety of organic compounds in CO2, water, and energy. We performed several experiments in collaboration with Sapienza University. We perform in our institute molecular ecology and biochemical analysis and some chemical analysis, in particular degradation patterns of organic contaminants. And the design of the microbial fuel cell was performed by Sapienza University. The capability of terrestrial microbial fuel cells to simulate natural microbial communities for the degradation of different persistent organic contaminants, adding organic amendments, was tested in several experiments. Here, as an example, we tested the capability of a terrestrial microbial fuel cell for promoting DDA degradation in soil in presence or absence of municipal soil waste compost. We also tested open and closed circuits of the microbial fuel cells. This system promoted a significant DDA removal after 2 months, while at the same time any pesticide decrease was observed in the batch condition. And compost addition stimulated microbial activity and improved the terrestrial microbial fuel cell performance for a longer time. We tested also the terrestrial microbial fuel cell only in the presence of the compost, and the structure and functioning of the microbial communities at cathode, anode, and bulk soil was studied and compared. Here you can see some outputs of the microbial fuel cells, and you can see that the compost addition enhanced the power generation and increased the open circuit voltage from the 3rd day of the experiment. And the power produced by the terrestrial microbial fuel cells with compost was constant constant up to the 5th day of the test. Then the performance dropped. In this slide, you can see that we analyzed the microbial community structure, and in presence of compost, an increase in Bacillus, Clostridia, Bacteroideia, and Delta Protobacteria was observed. But the important was the analysis of the genera that we did in different parts of the microbial fuel cells. And we found that Geobacter, that is a classical bacteria involved in the transfer of electrochemicals, was found at the anode, confirming its role in electricity production. So, nature-based solutions are useful tools for eco-friendly recovery of contaminated environments. They are in line with the circular economy approach, and they're also in agreement with the Sustainable Development Goals. And natural microbial communities are an effective tool for nature-based solutions. And thank you for your attention. I would like also to thank Professor Nicholas for involving me in this special issue, and also the people from MGPI and Dr. Zhao. Thank you.
Thank you, Dr. Grenny. That was wonderful. I'm sitting here excited about the whole. First of all, I've never been on a webinar or a program where everybody was right on time. So I think that's pretty amazing in itself. And secondly, I find myself fascinated by potential from both those talks. We are open for questions and answers. There's no questions being posted right now in the Q&A box, but we do have a question that was proposed in the chat. And let me start with that. And then I'll ask anybody out there, if you have questions, please now put them in the Q&A box, and we'll deal with them one at a time. Otherwise, I've got lots of questions that I'd like to get answers to from my two colleagues here, because I'm excited. So this question is from Deepak Kumar Yadav, and excuse me if I screw up anybody's names, because it's just the way it is. And he says, I am working on the mapping soil compaction, for which knowing soil moisture is paramount. Which type of sensor would be good for mapping soil moisture? Number one. Are there any database which gives real-time soil moisture data? Number two. So number one is, what would be a good sensor for mapping soil moisture?
Okay. Thank you very much for the question. And there are many soil moisture sensors available, and which one to choose is really depend on your research aim and the available resources to you also. And you can predict or measure the soil moisture from satellite imagery, but it's not real time. Usually 15 days, or it's also a coarse resolution from the satellite imagery if you want to use that for soil moisture prediction. And also this is the moisture for the surface soil layers. But if you're interested in the deeper soil depths beyond 30 centimeters, beyond 30 centimeters, you might need some other soil moisture sensors, such as the proximal sensors that, as I introduced, the electromagnetic induction. If you have a EMI sensor around and you know how to use it, it's a good tool for monitoring the soil moisture dynamics. Otherwise, there are also plenty soil moisture sensors you can use, which can be installed in the field and to monitor continuous soil moisture content, like every 30 minutes, and upload the soil moisture to the cloud. And you don't need to go to be on site. You can directly download the soil moisture information from the cloud. And the on-site installed soil moisture, such as the TDR, which enable you to install at different soil depths down to a few meters. And also there are capacitance probes available. And we are currently using a type of capacitance probe. It's permanently installed in the field for continuous monitoring soil moisture. It's called drilling and drop. It's very easy to install. It's from Santec company. And you just dig a hole and install the sensor into the soil. And it can reach down to 1.2 meters deep. And its accuracy quite good. And also neutron probes. You can use neutron probes to measure soil moisture in the field. Yeah, so it's, I think very much I answered the two questions.
The other one was databases which give real-time soil moisture data.
I think you have to create that database. I don't think there are some free and public available soil data moisture database. It's real time. You have to create your own, install your own permanent soil moisture and upload to the cloud to create your own real-time soil moisture database.
Okay, well, thank you. And Deepak, if you have any follow-ups, feel free to put them in the question box. We have another question, and this goes to Dr. Greni. This is Natalia Tachuk. Hope I said that right. And she says, thank you very much for a very interesting speech. Please tell the names of the electro-generated genus of bacteria.
Yes, thank you for the question. The genus of bacteria are Geobacter. And the very interesting thing that we found that this bacteria can not only produce electricity, so move the electrons, but also was able to degrade the contaminant. We also recently published an review on this. I can put the title in the chat if you want.
Okay, thank you. There are no other questions in the box, but I've got a couple. Dr. Chow, one thing that amazed me, the changes that have occurred in monitoring is your ability to go to soil depth. As a field soil scientist, I find that absolutely amazing. What kind of soil properties are available to get to soil depth through remote sensing?
From remote sensing, it's very difficult.
Yeah, so usually it's a surface. So you had electrical conductivity.
That's proximal sensing. This is close to the ground.
Okay.
Yes. So it's not from the satellite imagery. It's a proximal soil sensor. So you have to bring it to the field and to collect the soil responses across the field. And this a sensor you placed close to the ground, and you can drag across the field to collect the information, and then do need calibration to the soil properties. And this EMI.
Are we talking about ground penetrating radar?
No, it's electromagnetic induction.
Okay.
So it's emitting electromagnetic field to the soil, and then the conductors in the soil will respond to this magnetic field and generate an eddy current. And that eddy current will generate a secondary magnetic field in the soil. And then the sensor will receive that secondary magnetic field and calculate the ratio between the secondary and first magnetic field. And because this is the beauty of this EMI sensor is that it's able us to look at the deeper soil information. Not like I didn't introduce a lot of information on the remote sensing, the satellite imagery, is because it is looking at the surface soil layer around 0 to 30 centimeters. But proximal sensing, the ground penetrating radar, as you mentioned, and also this electromagnetic induction enable us to really looking at the hidden half below the ground. And yes, and they do need some geophysical inversion modeling to get to the information at each of the soil depths. Yes.
So is there a maximum depth and the maximum depth?
It's
And
depend
are
on
all
which,
soil properties? Pardon?
it depends on which sensor, which type of EM sensor you use. So the measurement depth is determined by the distance between the transmitter, which one transmitting the magnetic field, and the receiver. It depends on the it's a measurement depth is determined by the distance between the transmitter and the receiver. The far the transmitter and the receiver, the deeper you go. So there are many different EMI sensors available for the agriculture use. We usually use EM38 and Julian EMI sensors. It's usually can go to from 0 to 3 meters as the root zones. And if you want to use at some other hydrological, to predict the soil hydrological properties like the water tables, you might need to use other EM sensor like EM34, which measures a soil depth down to 30 meters. Yes, you can choose which EM sensor to use based on the research aim.
Are there soil physical properties that are appropriate for that, and then some that are not?
Yes, yes. Some are not, some are good. So it depends on it's a complex relationship between the EM response and the soil property, because there are multiple soil properties like salt content and soil texture and moisture. So it's a complex relationship. And usually if the soil is saline, and the EM response can be used to predict the soil salinity. And
okay, now
and
we
you
have a
can
question.
yep.
We have a question that might be somewhat related to see if so you tell me if you've already answered this question or not. It's from Fedele Caplari, and he says, please could you give a little more explanation on how you use the remote sensing to obtain the EC data? And I think you're talking about
Also,
proximal
this question is not remote sensing, it's proximal sensing.
Okay.
It's not from the satellite imagery, it's from the proximal electromagnetic induction sensing.
Right, and I think in our discussion, it seems to me you have answered that question. Would you agree?
Yes, exactly.
Okay. Okay, well, thank you for the question. Continuing the discussion, Dr. Godeni, you with your expertise in pharmaceuticals, I know you have interest in organic matter additions to soils and what might be in that organic matter. Antibiotics in particular. What is going to be the long-term effect of putting manure into soils if we don't take care of the antibiotics before we go in? I know this is an area that you've worked in.
So the addition of manure to soil can, of course, improve the soil quality in the sense of organic matter. But as you know, the animals are treated with antibiotics, so some residues of antibiotics can be also in the manure. And not only some residues of antibiotics, but also the resistant genes. So the problem of the application of the manure to soil can be the spread of the antibiotic resistant genes. So we performed some experiments in which we found that, for example, it is better not to apply manure, but for example, compost or digestate that are that can contain less amount of antibiotics and less amount of antibiotic resistant genes. So this was our mainly results that we had from several experiments. So it is important to consider not only the amount of organic matter for a soil, for enhancing the organic matter to the application of different amendments, but the type of the amendments that you have that you can apply. So you have to be careful for this.
Right. So antibiotics are given to livestock, right? But hormones are also given to livestock. Are hormones in manure a potential problem?
Sorry, I don't understand.
Oh, livestock antibiotics are given to livestock, and then that's what shows up in the manure, correct? So another thing that's given to livestock, which people have been concerned about, are the hormones that are given to livestock. Would hormones also be in the manure, and would they be a potential problem?
Not in manure, but probably in the Swiss sludge that can be applied to the soil. Swiss sludge that are from wastewater treatment plants. So I think that the main source of hormones can be from the biosolids from wastewater treatment plant, because the hormones can be from the humans mainly because they can use the hormones for them. And in the manure, I think I don't know, because in Italy we don't know, we didn't use the hormones for the animals. I don't know in other countries. But in any case, I think that biosolids can be more, they can contain more hormones.
Okay, great. Mara, what's our timing? I'd like to open it to my colleagues if they wanted to answer any questions, but what's the timing here?
Sure, we have 6 more minutes and then we can close.
Okay.
I have a question for Dr. Zhao, if it is possible.
Yep.
Yes, we got time.
It's possible, in your opinion, also to determine some parameters related to microbial community in the soil with your tools?
Yes.
I don't know.
Yes, I think it is possible because the microorganisms in the soil, I think it's definitely will influence some soil properties, like maybe more microorganisms, and the soil is able to hold more soil, more moisture, or it can change the soil structure or something like that, and then that can, if the soil properties or the soil structure has been changed, it for sure can be reflected in the visible near infrared spectral, the soil spectral reflectance, and also if the soil structure changed, it can be reflected also in the conductivity, the EMI responses, because the bulk density of soil changed, and that also influenced the soil conductivity. Yes, my answer is yes. Yes, and I think the sensor has the potential. I haven't tried, but it has the potential to be able to be used to characterize the soil micro organisms in the soil. And I know some teams in Australia, they are mapping the soil biodiversity or microorganisms at national scale. They are using different sensing technologies but they are mapping the surface soil layer as the satellite imagery just able to characterize the above half meter is the maximum. Yeah.
Okay, thank you so much. I think it is important to combine several tools in order to see the real quality of soil. No?
Yeah, the data fusion is combining different sensing techniques, and because each sensor sometimes works, sometimes not, and it's better to combine different sensors and to further improve the prediction ability. Yeah.
That might be the future, because at this conference I was just at, there was quite a bit of work talking about stacking models in terms of trying to improve the
Model
accuracy.
everything.
Yeah. So I think we're pretty much getting to the end of our seminar. I want to take the opportunity to thank my colleagues. I'm leaving a much smarter person than when I came to this seminar. I particularly want to thank our hosts. They've been excellent in trying to get us prepared for this and overcome all of our limitations in making this seminar available to folks. And with that, I think I'll turn it over to Tamara to wrap up.
Thank you very much. Before we wrap up, I just want to take a moment and extend the huge thank you to our incredible speakers. Your presentations today were both very insightful and engaging, and the depth of knowledge you shared was truly valuable for each of us. We learned a lot, and your expertise really made this event very memorable. I would like also to thank to our participants for being engaged throughout the webinar. Your questions and interactions made this experience even more enriching, and we are so grateful for your active involvement. Regarding the certificates, I saw that you write in the group. We will send the certificates a notification when we'll be ready. The certificates of attendance after the webinar is done will be generated, and we will inform each of you. Thank you very much to everyone for making this event such a success. Have a great day. Thank you.
Bye-bye, thank you.
Thank you very much.